Hello, our team is addressing racial disparities in corneal implants for presbyopia. I'm Cecilia.
I'm Michael.
I'm Brian.
I'm Samantha. So presbyopia is an increasingly global condition that is the gradual loss of one's eye ability to focus on near objects due to the loss of elasticity of the lens in the eye. Uncorrected presbyopia is very impactful on the quality of life. Despite many people worldwide suffering from this condition, the mechanism that causes presbyopia is still undetermined. This gap in research allows for further improvement on current treatments and management solutions. On top of this, it has been found that there is a lack of patient diversity in the clinical trials of several devices, which leads to the lack of confidence in the device's safety and effectiveness for certain demographics. One of these devices investigated is the camera inlay, which is an intraocular implant that can be improved significantly. The recent increases in aging population has resulted in the need for healthcare professionals to manage this condition in an effective manner. In 2015, it was estimated that 1.8 billion people globally had presbyopia, but it was expected to peak at approximately 2.1 billion in 2030. There are treatment options in the form of intraocular implants that range from $7,000 to $8,000. However, all currently available treatment methods accept some compromise in the quality and flexibility of vision at the different distances and do not account for the different eye shape across different races. The current treatments on the market for presbyopia are mainly bifocal lenses, intraocular implants, and inlays, such as the camera, raindrop, and FlexiView microlens. About 20% of individuals who receive the camera implant will request the removal of it due to dissatisfaction with the results. While camera is the sole large FDA-approved corneal inlay, that risk can be considered pretty high for the cost of about $7,000 to $8,000. Other cheaper options, such as reading glasses, prescription lenses, and contact lenses, may appeal to some patients, but face the same problem as the camera inlay, where the patients will be testing predetermined standards. The biggest limitation with the current treatments for presbyopia is the inability to ensure comfort and usability for all people, specifically across different races. The comfort cannot be ensured for all customers, as it still may be considered a foreign body in the eye. Additionally, it cannot correct long distance vision and require some amount of surgical expertise to perform the implanting of the device. The main objectives of our intraocular implant design is to reduce the rejection rate of the implant due to discomfort and the implant being made without taking all eye shapes and sizes into account. One specific study investigating the camera inlay competitor shows how different racial backgrounds have significantly different outcomes using the product due to the shape of the cornea among racial groups. Since ocular anatomy isn't the same for all ethnic backgrounds, it is important for our device to address this by being adjustable and personalized for the patient. Our goal is to make this medical device more attractive as a treatment option for lifelong solutions of presbyopia, which affect over 1 billion people worldwide. Another way we wish to achieve this is to significantly reduce the cost by using other materials and having insurance companies cover a large part of the cost. By improving the overall success rate and usability across different races, we hope to make the intraocular implant a more attractive lifelong solution to presbyopia. Our design will be modeled after the camera inlay, which uses the pinhole effect in order to improve vision by only allowing focused light to pass through a small hole in the device. It will be a lens with a small ring made of PVDF that is implanted on the cornea through a surgical procedure, where a femtosecond laser is used to create a pocket in the patient's non-dominant eye. Once it is correctly placed, the patient should notice improvement in their vision as soon as 24 to 48 hours. We'll also be adding a biomimetic phosphorylcholine coating to ensure minimal adhesion of cells to the material surface and promote proliferation. This PC coating will also aid in minimizing the formation of giant cell deposits on the lens of the implant, which improves the overall vision of the patient as well. One of the main areas of improvement we want to address with our design is the idea of customizable dimensions for the intraocular implant. There are several studies that show how ocular anatomy differs between ethnic backgrounds and can therefore cause complications with the corneal implants currently on the market. The ability to customize the interior and exterior diameters, as well as the height from the pupil surface of the implant, will help all patients who suffer from presbyopia benefit from our device.
Our design will be made using the simple yet durable and relatively inert biomaterials polyvinyl iodine fluoride with a coating of collagen 1. The PVDF's thermal and chemical degradation properties, as well as its permeability, allow the implant to perform well in the corneal environment. It has high abrasion resistance and low water absorption, which are key to ensuring the comfort of the patient. The PVDF is easily manufactured and modified, making it an easy choice for a material with its complementary mechanical properties. The modification of collagen 1 coating will promote corneal epithelial cell proliferation and adhesion. Thus, the cells will adhere and turn in and in turn accept and incorporate the implant into the cornea. Both PVDF and the collagen one are slightly expensive. The cost of the implant and the surgery will most likely be covered by the insurance plans. Each design will be tested and modified to fit the specific person's request by using basic eye tests to determine the severity of the patient's presbyopia, and the spherical dome model will be used to find the corneal dimensions. The spherical dome model will allow us to attain information about the cornea, such as the stress, intraocular pressure, thickness, diameter, and anisotropy factor ranging from 0 to 1. These tests will then be utilized to create the perfect fit for the patient and their needs, providing a functional inlay, as all these factors will differ from patient to patient. The pinhole technique will be used to create these inlays, where a small opening of the inlay will allow for light to be focused in one area as it passes through the cornea, allowing for a clearer image to then be formed. The implant must have certain mechanical properties in order to suffice the needs of the patient, as well as the safety and comfort. The implant materials need to show good abrasion resistance and low water absorptions, which is crucial to the comfort of the patient. The implant must be able to undergo the basic stresses and strains within the cornea without failure. The material must also be able to withstand the average bodily temperature range and any chemicals or proteins that may be found within the cornea to lessen any chance of degradation, so the implant does not become deformed or damaged in any way, leading to the failure of the implant as well as the discomfort of the patient.
So it is important to understand the local and systemic inflammatory and immune responses in regards to our design to prevent rejection and failure. The tissue we plan to modify is the cornea. There is a relative immune privilege when working with the cornea due to the anterior chamber associated immune deviation, ACAD, which making it the corneal implants less prone to rejection than other organ transplants. That being said, disturbances in the ACAD increase the risk of rejection and must be taken into account. Contact with the vascular system can activate rejection as host immune cells recognize a foreign body. In order to prevent this, corneal neovascularization measures will take place pre and or post-operatively, as close proximity between vasculature and implant increases the probability of an immune reaction. Inflammatory diseases such as interstitial or traumatic keratitis carry a high risk for endothelial rejection. Type 1 helper T cells, Th1, is considered to be the main cause of the rejection process. Other systemic immune competent cells are able to recognize endothelial antigens circulating in the interior chamber and may result in localized non-ocular infections due to the increased amount of circulating acute inflammatory mediators. Local or systemic steroids may be used to reduce the risk of implant rejection. However, we plan to lessen these risks with our thorough biocompatibility assays. In order to test for biocompatibility, in vitro assays were done using PVDF with and without the PC coating against the materials controls of silicone hydrogel and the conventional hydrogel lenses. All lens tests were performed under sterile conditions and tested in 3 states: no treatment, PBS rinsed, and PBS soaked. In vitro studies were conducted in 2 phases. The first phase tests for absorption of fibrinogen through ELISA and determines cell adhesion with light microscopy and image analysis, as well as determining bacterial adhesion using similar techniques to the cell adhesion test. The second phase of the in vitro assays will focus on testing for proliferation on the surface of the materials and stain for macrophages. This phase will also test for granulocyte adhesion and activation. Finally, corneal endothelial touch tests will be performed to test the degree of damage using force. In vivo testing will be conducted on small and large in vivo using mice and cows respectively. All animal studies will be tested in accordance with the Animal Welfare Act, as well as any other federal laws addressing the standard of care of animals at research facilities. For both small and large models, the animals will be anesthetized and the lens will be removed from only one eye. Our design would then be implanted in one eye and sutured using a single nylon suture. Once the design has been implanted, the animals will be allowed to roam free and will be monitored on a daily basis. At 1 week, 2 week, and 4 weeks, the inflammatory response regarding interior flare and anterior cells will be assessed by slit lamp examination. Finally, long-term studies beyond 8 weeks will also be assessed. To ensure data reliability, a minimum of 2 observers will assess the animals and blind score them. We will use a minimum of 6 animals per group using the untouched eye as control. For the in vitro studies, it is expected that there is a greater absorption of fibrinogen, better adhesion of fibroblasts, lens epithelial cells, and macrophages, as well as an increased activation of granulocytes and are designed when compared to controls. In regards to the PC-coated materials, we expect that the in vitro studies further suggest improved ocular compatibility. The PC coating will especially help minimize the formation of giant cell deposits on the lens surface by minimizing this initial adhesion of diffuse cells to the surface material. We expect the in vivo studies to confirm the results of the in vitro studies. There should be no differences observed between the postoperative interior flare or interior cells for our lenses. In addition, no bacterial growth or corneal endothelial damage should be observed in any of the animals. Our thorough biological screens have been intentionally designed to fully assess the potential of our biomaterials for this application.
The process of inserting our inlay into the eye would be very similar to other eye operations that would be performed by a surgeon. First, a femtosecond laser will be used to cut open the cornea, the first few layers of the cornea, and then the inlay will be positioned inside of the cornea, and then that flap will then be closed and the cornea will be allowed to heal on its own. As the healing is much dependent on the stem cells as well as the basement membrane below, it's very important for the material to not hinder this process of healing, as there's not much that any of the materials can do to expedite this process. So it's very important that we choose a material with the proper biocompatibility for this application. In order to manufacture the lens, we will, after receiving the measurements, as Brian touched on earlier, we will use a laser cutter to cut a disc of the PVDF material, and then laser cut small incisions inside of that so that it becomes even more porous and allows for oxygen and other important nutrients to move properly throughout the eye and facilitate the healing. Then the PC coating will be applied to the top of the all over the lens, and then it will be sterilized using ethylene oxide gas at a low temperature, and then double sterilized package to not be opened until it is ready to be used. All of our design specifications were according to the ISO standards of 10993, which is specific for intraocular lenses. There are still some potential issues with our design. There is always the potential for rejection. While this is a very minimal chance with the steps we took with biocompatibility, with the PC coating and the materials, there is still always the potential for the body to recognize the implant as a foreign body and then spark an inflammatory or an immune response. There's also the potential for decreased performance over time due to degradation. For the camera inlay, about 20% of the patients after 4 years saw a decrease in performance. However, there's also other studies saying that the materials and the inlay itself should last a lifetime without any performance changes. And the last issue would be the surgical expertise and symptoms, where the proper handling of the inlay itself, as well as the conduction of the surgery, will greatly impact the recovery time for the patient. Thank you for your attention. Do you have any questions?